So let's move on to the idea that with Mendel's dihybrid cross showed that each pair of alleles segregates independently of the others during gamete formation. So what that means is if we look at the parents here in this dihybrid cross represented by the P's, this individual is homozygous dominant. Right? Both of the alleles are the same and they're dominant because they're capital. And the allele, which is genetic information, right, a version of a gene, codes for the shape of the P, right, round versus wrinkled. And this individual, it's lowercase r and r, so it's homozygous recessive for the shape alleles, shape of the P. And back to this one, it's yellow, so that's the dominant color, homozygous dominant, 2 capital Y's. This individual is green, which is the recessive trait, characteristic, right, what we see physically, and that will only happen if it's homozygous recessive, 2 lowercase y's. But when the gametes are formed, the R allele will go into the gametes completely independently of the Y allele. Now in this individual, it only has the dominant capital R, so the gamete can only receive that particular allele. And the same with the dominant Y allele. It can only put or have a dominant Y allele go into its gametes. And the same thing for this one, which is homozygous recessive for both alleles. Okay? But notice the offspring. The offspring gets dominant and recessive allele for shape and dominant recessive allele for color. So the individual, the F1 individual, this is the parental cross, and that's the F1 individual, F1 generation. It is heterozygous for both alleles, meaning heterozygous, one of each. Dominant recessive. Since it is genetically heterozygous for dominant and recessive alleles, the dominant trait is what is seen physically. So R capital for round and Y capital for the yellow color. Now genetically, it does have the genetic information for the wrinkled shape of the P and the green color, but it is recessive characteristic or trait, so it is not seen in the F1 generation. Then down here, this is the results of the F1 cross, meaning one individual that's heterozygote for both alleles would be crossed with another same heterozygote individual for both alleles. Now you notice that when this one is going to make its gametes, the gamete can now have either a capital R or a lowercase r. And the gamete can have either a capital Y or a lowercase y. So this is the 4 possible gametes made by this individual. Okay? And notice there's a 25% chance, 1 in 4, of a particular combination of alleles in that gamete. And the same thing for the other individual because it's an F1 cross. Then when the gametes fuse, these are the potential genetics, meaning the genotypes of the individuals, and the appearance of them would be the physical characteristic, the phenotype. And the phenotype is always determined by the genotype, right? The physical characteristic is always determined by the genetic characteristics or the genetics of the individual. Okay? So that's what's referred to as the alleles segregate independently during gamete formation. And this is somewhat complicated when we first examine it, but again, you're not responsible for knowing the results of a dihybrid cross, just the simpler monohybrid cross that we saw earlier, right? So the inheritance of one characteristic or character has no effect on the inheritance of the other, right? At least at this point. Okay, so this is what's called Mendel's law of independent assortment, right, of the alleles during gamete formation. Now remember, the alleles are carried on chromosomes, so that applies to the chromosomes, law of independent assortment. So your textbook mentions how you can do a test cross to determine an unknown genotype. So the example here are labs, right? Labrador retrievers, right? The dogs we call labs. So the black coloration on the coat is the dominant phenotype. That means that the dog, genetically, its genotype has to have at least one capital B. It can have two capital Bs, right? The black dog. It can be homozygous dominant, or the black dog can be heterozygous, a capital and a lowercase, a dominant and recessive allele. Because since black is, doesn't mention it here, since black is the dominant phenotype, right, the coloration, what you see physically, the genetics of the individual only has to have one dominant allele. If it has both dominant alleles, it's homozygous dominant. It can only be black. But if it has one dominant allele and one recessive allele, the individual is heterozygous. But since the black color is dominant, excuse me, the individual is going to be black in coloration, right? His fur or her fur coat. Now, the chocolate lab, the brown coloration is recessive. So that can only be possible, the phenotype, it can only be brown or chocolate if the dog genetically is homozygous recessive, right? Meaning there's no dominant allele to get expressed. There's no dominant allele to hide or mask the expression of the recessive characteristic. Okay, so you have a chocolate lab. It mates with the black lab. The chocolate lab we know has to be homozygous recessive. Otherwise, it's not going to be chocolate or brown. So the black lab, if you don't know its genotype, well, you can do a genetic cross. You can mate these two and examine the offspring. Okay, so there's two possible genotypes for the black lab. Its gametes are going to be it's either capital B, homozygous dominant, which means its offspring. It means its offspring are going to be determined by its gametes, and it can only produce a gamete with the capital B, the dominant allele. If it mates with the chocolate lab, the chocolate lab is homozygous recessive. It can only put a recessive allele in its gametes. When they mate, fuse, the gametes, right, egg and sperm, you get the baby individual, puppy dog is born, it's going to be black. Why? Because the individual is heterozygote. Okay? However, if the black lab mates with the chocolate lab, again, the chocolate lab can only produce gametes with the recessive allele, but the black lab, if it is heterozygote, it can produce both. It can produce gametes with one dominant allele or one recessive allele. There's a 50-50 chance of this happening with the gametes it produces. So they have a litter, and as you know, dogs usually have more than one baby. They have 4 or 6 puppies, whatever. Let's say they have 2, and one is black and one is brown. Aha! The only way the brown will come up is if the black lab contributed a recessive allele. So if you do have a chocolate lab puppy, that means the black parent has to be heterozygote. Okay? Now, ideally, or if you were actually going to do this experiment, you'd have a couple of litters to make sure you had enough offspring to verify that, oh yeah, at least some chocolate labs would pop up to indicate that that black lab parent had to be heterozygote. So when we're talking about these genotypic crosses and such and mating events, this takes some time for students to absorb and understand and be able to answer questions on quizzes or exams. So it is very beneficial for you guys to pull out a piece of paper and write this stuff out. Don't just simply read it once or listen to it once. It takes time, right? Genetics is not super easy. It's a bit challenging sometimes. Okay, so I mentioned phenotype and genotype. The phenotype, think physical, pheno, right? PH. The phenotype is the appearance of the organism. This dog is brown chocolate. This dog is black. That is it. Those are the organism's phenotypes. the phenotypes is based on the genetic expression. So that's the genotype, the organism's genetic makeup. What alleles, right, genes does it have on its chromosomes? Because the genotype determines the phenotype. Because the genotype has alleles. Alleles are variations of genes that code for particular proteins, in this case proteins that are in the fur that make the hair black or chocolatey brown. Okay? Just like you, if you have black hair or brown hair, it's not because you colored it, which you could, of course, but your hair is naturally black or brown or some other color because you have alleles that code for the colors that are incorporated into our hair, which gives our hair a particular color. The same thing with your eye color or skin color, those sorts of things. It's all based on our genotype. So rules of probability. So genetic crosses obey rules of probability because, as we talked about, what we look like, the phenotype is based on the potential gametes that are formed, the genotypes. And as we'll see here, it's based on the probability of making any particular type of gamete and the probability of any particular gametes fusing to make the genotype of the individuals. Okay? So it's like tossing a coin or drawing a card. If you have a coin and I say toss it once, there's a 50-50 chance, 1 in 2 chance it's going to be head or tails. Okay? If you toss it twice, what's the probability of it being heads twice in a row? It's actually 1 in 4, as we'll talk about here. Okay? So the rules of multiplication based on the rules of probability. So the probability of a compound event is the product of the separate probabilities of the independent events. It's like, what? Okay, what this means is, let's look at these potential individuals. Now they're using coins here, but you can go back to look at this chart, for example, or even this one here, to determine or to be able to relate this to gamete formation. So individual male, right? If the male is heterozygote, the male can make sperm. The male is diploid, right? Has both copies of the alleles because they're on homologous chromosomes that male inherited from its mom and dad. Okay, so when the male produces sperm, you go from diploid to the haploid situation. Only one of the alleles is going to go into a gamete, and the other alleles will go into other gametes, right? Because the chromosomes and the alleles are on chromosomes separate during meiosis. Right? Go back and look at mitosis and meiosis, particularly meiosis here, right? So there's a 50-50 chance, 50% chance that any particular male gamete, a sperm, is going to contain a dominant allele or a recessive allele in this particular case. If the female is also heterozygote, same thing. There's a 50-50 chance that her eggs are going to have the recessive, or the dominant allele. That should be pretty straightforward, okay? Because there's only 2 alleles. They're different, of course, so there's a 50% chance that her gamete, an egg, will have the dominant or the recessive allele. Okay, so now they mate. Sperm fertilizes egg. If that happens, what's the probability that the egg is going to be homozygous, that the offspring, the baby that's produced, right, that's genotype, what's the probability that its genotype is going to be homozygous dominant, capital B, capital B? Well, it's going to be the 1/2 times the 1/2 or 1/4, meaning 25% chance. Heterozygote, 25% chance. Heterozygote, 25% chance. Homozygous recessive, 25% chance. If just look at the Punnett square here, 1, 2, 3, 4 squares, it's like there's a 25% chance this will happen, a 25% chance this will happen. But notice with the heterozygote, it can be capital lowercase b matching those gametes, or lowercase b capital B matching those gametes, which means there's actually 25%, there's actually a 50% chance of being heterozygote. Again, you're going to have to spend some time looking at this and writing this out on your own to be able to get comfortable with these probabilities and such, and how the gametes form. Okay? We'll continue on the next lecture part.